Today, we're diving into one of the biggest scientific breakthroughs of our time. But this story doesn't start in some fancy, high-tech lab. Nope. It starts with an ancient, invisible war that's raging all around us, in every drop of water.
See, it all kicks off with a mystery that had scientists scratching their heads for years. How on earth can a simple, single-celled organism possibly have a memory?
Well, it turns out bacteria are in this constant life-or-death struggle against viruses, these things called bacteriophages that hunt them down. And to survive, they've evolved this incredibly clever immune system. And this natural defense system, it's called CRISPR. The best way to think about it is like a genetic most wanted list. Every time a new virus attacks, the bacterium literally snatches a mugshot, a piece of its DNA, and keeps it on file forever. And it works in this really elegant three-step process. First step is acquisition. The bacterium grabs that little piece of viral DNA. Then, expression. It weaves that snippet right into its own genetic code for safekeeping. And finally, interference. It makes copies of that viral code that acts like a guide, ready to spot and destroy that virus if it ever shows its face again. So the bacterium is basically vaccinating itself. And here's the kicker. Because this most wanted list is written directly into its DNA, it passes that immunity down to all its kids. It's a perfect inheritable defense system. Okay, so you've got this amazingly precise natural system. And this is where the story takes a huge turn. Scientists looked at this and asked a question that would change, well, everything.
That was the massive aha moment, the big realization. What if we could hijack this system? What if we could give it our own most wanted list and point it not at a virus, but at literally any gene in any organism we choose?
And that is exactly what they figured out how to do. Scientists managed to isolate the two key parts. You've got the Cas9 enzyme, think of it as a pair of molecular scissors, and then you have the guide RNA, which is like a biological GPS, telling those scissors the exact genetic address to go to and make a cut.
Now, this might look a little technical, but the key idea is right here. That little purple strand, the guide RNA, is the brains of the operation. It leads the big green Cas9 protein right to its target on the DNA, getting it ready to make a perfectly clean cut.
And this simple system became the foundation for a genetic editing tool that is just worlds better, more powerful, way cheaper, and so much easier to use than anything that had ever existed before. You see, before CRISPR, we had other tools, but they were incredibly clunky and super expensive. For every single gene you wanted to edit, you had to build a whole new, complicated protein. It was a ton of work. With CRISPR, all you have to do is make a new guide RNA, which is small, cheap, and really simple to design. It basically put gene editing into the hands of almost any scientist anywhere. But listen, the cut itself is only half the story. The real magic is what happens next. As soon as Cas9 snips the DNA, the cell kind of panics and fires up its own natural DNA repair systems. And it's those repair systems that we can cleverly hijack to do the editing for us. The first repair pathway is like the cell's emergency response team. It's called NEJ, and it basically just rushes in and stitches the broken DNA back together. Now, it's often a bit messy and it makes little mistakes, but those mistakes are actually super useful if you just want to knock out or turn off a faulty gene. But this second pathway, HDR, this is where the true power of gene editing comes in. With this one, we can supply the cell with a new, custom-designed piece of DNA. The cell's repair machinery sees our template and uses it to perfectly rewrite the genetic code right where the cut was made. It's the biological version of find and replace on your computer. And as you can imagine, this ability to precisely find, cut, and even replace genetic code has just unleashed this incredible wave of new possibilities across all of biology. I mean, just look at medicine. The potential is staggering. Researchers have already used CRISPR in the lab to correct the genetic mutation for cystic fibrosis in human cells, to fix a gene that causes cataracts in mice, and to create better animal models to study complex human diseases. And for pure, basic science, oh man, it's like we've been handed a universal key to the genome. Scientists can now tweak multiple genes at once, tag proteins to literally watch them move around inside a living cell, and systematically turn off thousands of genes one by one to finally figure out what they all do. You know, one of the early papers on this, way back in 2014, summed it up perfectly.
They said CRISPR took gene editing from just being a technical possibility and turned it into a practical reality for pretty much any lab in the world.
And just when you think the story can't get any cooler, it takes another twist. Scientists got clever and actually broke the scissors part of the Cas9 enzyme. They created a version that can't cut DNA anymore, but it still has that perfect GPS. So this dead Cas9, or dCas9, has become this amazing programmable delivery drone. We can attach other things to it, like a light switch to turn genes on or off, or a tiny fluorescent beacon to make specific parts of a chromosome literally light up under a microscope. And it does all this without changing a single letter of the DNA code.
So let's just step back for a second. We've journeyed all the way from a microbe's ancient defense system, something that was hidden in plain sight for billions of years, to a tool that is fundamentally rewriting biology as we know it.
What started as a simple, elegant way for bacteria to remember their enemies has given us, humanity, this unbelievable power to understand, and maybe one day correct, our very own genetic code. This technology brings with it just immense promise, but also some really profound questions for our future. Now that we have the power to rewrite the book of life, the biggest challenge ahead might be figuring out what rules we should write for ourselves. Thanks for joining this explainer.